We will be showing you how science has confirmed the Bible. Science has discovered the first man, and that first man lines up perfectly with biblical origins.
And they've done this one time in a gene in mice and in fruit flies, and they call it the fountain of youth. And in mice, just editing one gene allowed them to live 23% longer. And Dr. Valtier said with just a small modification of the genome, you can go from 2 years life expectancy to 100.
The model Matt and I, we are in the affirmative tonight that we are arguing for, defending is the separate ancestry model.
What in the world does that sound familiar to anybody? What? Perhaps a global flood?
And that's why we say life could not have evolved through evolutionary processes such as selection, which is a fine-tuning mechanism that keeps a species as strong as it can be. It's helpful, but it's not going to drive fish to fisherman, bacteria-like organism to biologist evolution over time.
I was going to go through more of those studies, but no problem. We have the observed rate way up here with the estimation rates of evolution way down here. They don't match. One's an assumption, one's viewed, one's falsified. All the top geneticists completely agree with this.
Man, I'm pumped for this. Debate seminar, Adam, Eve, and biblical creation. We are taking on Culture Catz, who I do highly respect. I think this is going to be good. This is going to be one to remember. Resolution. Adam, Eve, and biblical creation are true. Donny and Matt Naylor, we take the affirmative. Culture Catz is taking the negative. Here's our website, StanfordTruthMinistries.com, where you can find endless content on the topic of origins. I've written several books. Here are the most relevant ones to tonight's topic. This is Special Creation, where we touch on all of these same topics that we'll be discussing here tonight. The Endogenous Retrovirus Handbook. I wrote this book in response to the so-called best evidence for evolution, shared endogenous retroviral-like sequences in the biological world, specifically focusing on humans and chimpanzees, the great apes. In this book, I demonstrate that the best understanding of ERVs is that they are mostly created units of DNA function, as we've discovered incredible functional roles associated with ERVs. They function in embryological development. They work to determine cell types. They act in cell stress responses in the immune system. As a matter of fact, we wouldn't exist without them. You can get that book and the others on Amazon. Here's an article that I co-authored with Chris Roop. This was reviewed for us in a rigorous review process by Dr. John Sanford, author of Genetic Entropy. In this, we talk about fixation equilibrium. The fact that the observed mutation rate, the neutral mutation rate, even according to prominent evolutionary scientists like Fatuma, the neutral mutation rate equals or approximates the fixation rate. So the neutral mutation rate, as we will see here today, is fast. And those evolutionists that want to appeal to the substitution or fixation rate and say, well, that's a lot slower, we demonstrate here that no, the observed rate, which is fast, approximates the long-term substitution rate. Here's a couple articles that we did. We engaged in a research project looking for in the genome in relation to the human chromosome 2 argument. We searched the genome. Chris Root specifically did the work there using BLAST, and we discovered similar signatures in other chromosomes other than the chromosome 2, where you've got the telomeric repeats in forwards and in reverse, TTA, GGG, CCC, TAA, in mirror form. We found a large signature in the chromosome 9, suggesting that the human chromosome 2 might not be a fusion after all. But we can elaborate on that if necessary. Not sure if that's going to be brought up or not, but if it is, we're ready to discuss it. Overview, real quick overview. In this opening statement, Matt and I are going to discuss several significant topics. These topics include the Y chromosome. This is the piece of DNA that makes a man a man. We will be showing you how science has confirmed the Bible. Science has discovered the first man, and that first man lines up perfectly with biblical origins. All men in the world have inherited Adam's Y chromosome. We've literally discovered Adam and Eve as a matter of fact, in our genetics. This is why we will also be discussing that little piece of DNA that can be traced back to the mother of us all, Eve. This DNA is the mitochondrial DNA. This opening statement will answer this evening's resolution. Yes, Adam and Eve are real and biblical creation is true. We are going to be discussing the human genome tonight. This is going to be heavy on genetics. What is the genome? The human genome is all your DNA. You can see that you inherit chromosomes in pairs, one from mom, one from dad. Recombination takes place where you get the shuffling of pre-existing DNA variants leading to variation. That's why hypothetically a single couple could have 1 million kids and every kid would be different because we have what's called pre-existing heterozygosity. And so the genome is essentially the complete set of genetic instructions that a biological organism uses to develop and to function. Here you can see the XY, XX. If you're XY, you'd be a male. We're going to be focusing a lot on the Y chromosome. XX, you'd be a female. God is an amazing engineer. A human cell contains 6 feet of DNA. If all the DNA molecules were stretched out end to end. This is all packaged and organized into chromosomes that fit nicely into the tiny cell nucleus. If life were simple, evolution might have a chance. Life is not simple. Life is incredibly complex, which is why the most parsimonious explanation, the best answer to origins, would be special creation, which is our affirmative tonight, specifically the special creation described in the book of Genesis with Adam and Eve. Letters in the genome are incorporated in multiple messages. These are essentially the instructions for building and operating a living organism. A single sequence of DNA can be read in different ways to produce different proteins. And this all evolved over time randomly for no reason at all. We have these overlapping codes. For example, we write books, but we can't write books that can be read both forwards and backwards. DNA can do this. ATCG, every other letter, you could have a third encoded message. That's why the genome is both polyfunctional and polyconstrained, because you get a mutation in a sequence, you're going to mess up several things, not necessarily just one. And that's why we say life could not have evolved through evolutionary processes such as selection, which is a fine-tuning mechanism that keeps a species as strong as it can be. It's helpful, but it's not going to drive fish to fisherman, bacteria-like organism to biologist evolution over time. And mutations, which are primarily deleterious. Most mutations are low impact, selection can't see them, they build up. Can we design computers in a way that mimics the human genome's sophisticated organization that features what? Overlapping coding regions and regulatory elements? The answer is no. Yet this is what we see in the genome. Modern computer systems, which are highly intricate, lack the inherent complexity and adaptability of biological systems. If we can't design that which is present in the genome, that means the genome itself could not have evolved through naturalistic evolutionary processes. Catz and other evolutionists have quite the task. Why are we, Matt and I tonight, to believe that these incredibly sophisticated systems evolve through naturalistic evolutionary processes over time? Mutually interdependent systems. Why special creation is necessary in light of the data and the facts. I'll go over one. I could go over many, but we've only got time for one. DNA and DNA repair enzymes. It is said that in a single day, we in a single cell could experience up to 1 million DNA lesions or DNA breaks. We have 40 to 65 trillion cells. That's a lot of DNA mistakes in a single day. Thank God we have these amazingly designed DNA repair enzymes that go in and fix a lot of these DNA breaks. Now, yes, unfortunately, some do get passed on in the germ cell line. The mutation rate in autosomal nuclear DNA is roughly 100 new mutations per person per generation, but many of them are fixed through DNA repair enzymes. Now, here's the chicken and egg problem that the evolutionist has, and we'll see how our friend Catz here addresses this tonight. We have the DNA repair enzymes, the information for them are encoded in the DNA. You need both coming into existence at the same time, because if you have DNA without DNA repair enzymes, then DNA is not going to survive or thrive for very long. Will be extinct in a few generations. Now, if you have the DNA repair enzymes but no DNA, then you're not going to have the information necessary to encode for the DNA repair enzymes. That's why they both had to have come into existence at the same time, which is what we mean by mutually interdependent systems. Okay, let's continue on here. Here's a quote from a famous theistic evolutionist. These are the kinds of arguments that you'll see in an attempt to counter Adam and Eve, the existence of Adam and Eve. So this is Francis Collins, and he said, there is no way you can develop this level of variation between us from one or two ancestors. This argument assumes that all DNA variation is due to mutations. There are two very different positions or explanations for the origin of DNA variation coming from the creationists and evolutionists. As you can see here on the slide, the evolutionists explain the vast majority, if not all, DNA variation as a result of mutations over time. Creationists, we explain the vast majority of DNA variation as being the result of design diversity or created heterozygosity. That is the model that suggests God would have front-loaded Adam and Eve in the original created kinds with pre-existing DNA diversity that can be utilized through genetic mechanisms to produce variation quickly. Now, this model makes testable predictions. One testable prediction has to do with DNA function. And interestingly, ENCODE and various research projects have discovered that at least 80% of the genome has activity. This activity is not spurious. The reason why is because transcription is an energy-intensive and resource-intensive process. Every time you add a single nucleotide to a growing RNA chain, what you are consuming is energy, and you are using ATP. And so if most of this was junk or noise and not legit activity, beneficial activity, the 80%, well, then evolutionary mechanisms over time should have weeded out all of that excess genetic baggage, because it would be wasteful of resources and energy for the cell. And so the best explanation is that this 80% biochemical activity is functional activity, which is why it remains. And we have really good preliminary evidence for genome-wide functionality. We have evidence for functions in just about every single DNA unit or genetic marker that proponents of common descent point to as evidence for evolution, these so-called genetic scars, genetic mistakes, viral DNA, pseudogenes. Here's several papers going over the many functional roles for pseudogenes. They're necessary to sustain healthy life processes in the cell. Endogenous retroviruses, we've identified numerous functional roles for endogenous retroviruses. They're playing an antiviral role in the genome. Were any of these predicted by evolutionary scientists? I'd like to know that. I'd like to have an answer to that. They function in determining cell types and regulating gene expression. ERVs are essential in regulating early stages of human embryonic development. There's one paper that literally says if it were not for ERVs, and a very specific ERV, then we as mammals would not exist. I like to exist. Catz likes to exist. Matt likes to exist. Hopefully you in the audience like to exist. And so that's why common design is the best explanation for these incredible DNA units that we find littered in genome. Several more papers going over these functions. And so the design diversity hypothesis has some really good evidence. Okay, the biblical model of human origins. What does the Bible say? Well, really quickly, the Bible clearly teaches that man was created separate and special from any other form of life. Genesis 2:7, Genesis 21 to 22. God created two people, Adam and Eve. And the book of Genesis makes several genetic statements with significant implications. Jesus confirms this. At the beginning, he made them male and female, Mark 10:6, Matthew 19:4. 1 Corinthians 15:45, the first man, Adam. So he's described as being the first man. And Eve in Genesis 3:20, she's the mother of all living. It's pretty clear that the Bible is telling us there's no common ancestry between humans and any other form of life. The question is, do we have scientific evidence supporting that? The answer is yes. We've gone over just some of that so far, but in my last 15 minutes, we're going to go over some more. Overview of the evolutionary and biblical creation models. As any informed person knows, common descent here, this is a sophisticated phylogenetic tree of life, would suggest that all life, including plants, animals, so that would be you as a human related to a banana plant, going back to a single common ancestor. Right here, this is an evolutionary tree showing all life, fish, insects, chimpanzees, humans, algae, all related. The model Matt and I, we are in the affirmative tonight that we are arguing for, defending, is the separate ancestry model. So as you can see here, you've got humans in blue with your extinct human groups like Heidelbergensis, Neanderthals, Floresiensis, Naledi, so on and so forth. We can get into that if necessary. I suspect we'll be focusing mostly on genetics tonight, that humans are separate from any other form of life. I like sticking as close to home as possible. I don't have to show you why humans and strawberries aren't related through common ancestry. I can show you why humans and chimpanzees aren't related through common ancestry. Chimpanzees are said to be our closest cousin. We're supposedly, we go back to a common ancestor roughly 6 to 9 million years ago. If I can show you chimpanzees and humans aren't related, then guess what? Strawberries and humans are definitely not related. So we'll stick as close to home as possible tonight. The evolution and creation models, they have a lot of overlap. What I mean by that is there's a lot of agnostic evidence. Okay, there's pieces of evidence that both models can largely account for. If I were to say, Catz, the sky is blue or the earth is round, okay, as we can see from his background, he's definitely right on that. He would say, well, that doesn't just mean creation's true, because the evolutionary model can also explain why the earth is round and the sky is blue. That's agnostic. That's what's called non-discriminatory evidence. There's a lot of overlap where we have pieces of evidence and observations that both models can account for. Okay. Homology, these shared structures in the biological world. Catz would say that those were inherited by past common ancestors. Okay. You've got variation over time, adaptation. So these structures modify, descent with modification. We as creationists, we would say human engineers design in homologous patterns. Why shouldn't God? What about nested hierarchies? Okay, humans are a lot more similar genetically, physiologically, morphologically, in anatomy with humans. They got more in common with chimpanzees than they do with monkeys, fish, and banana plants. Okay, human engineers also design things in nested hierarchical patterns. A sedan has a lot more in common with a hatchback than it does with a boat or a skateboard. And so these are not very helpful. It is interesting, though, in the world of homology and in the world of nested hierarchies in DNA, I wonder why, if common descent is true, why do we have such strong nested hierarchical signatures? Because apparently if we've been accumulating all these mutations for millions of years and most of them are neutral, and it's that neutral variation that selection acts upon to drive evolutionary change, it's interesting that we have such strong signatures, because you think those mutations would disrupt those signatures over time if common descent were true. And so I'd love to see a source or reference that shows evolutionary scientists. I'm not saying it doesn't exist. I'm just interested to see if evolutionary scientists predicted ahead of time homology and nested hierarchies in DNA. Perhaps Catz could provide that. The human germline mutation rate. So selection apparently acts upon genetic variation. The mutation rate is high, but unfortunately for evolutionists, the vast majority of mutations are deleterious. They're damaging, they're disease causing, and selection can't see them, so they build up. Adam and Eve predictions. Low genetic diversity, one Y chromosome ancestor, one mitochondrial DNA ancestor. We're going to be discussing these a lot tonight. Matt also has his roughly 25 minutes to get into these. Low genetic diversity. Okay, you've got two models, the biblical creation model and the evolutionary model. In the Bible, it's clear. God creates two people just roughly 6,000 years ago, Adam and Eve. What that would do is restrict levels of genetic diversity. You wouldn't have a whole lot of genes floating around a population of two. But if evolution were true, and we've been evolving for, let's say, hundreds of thousands of years, going back to the out of Africa scenario, somewhere between 100 and 200,000 years. Mutations add something new because they're adding something that was not previously there. It doesn't necessarily mean that that which mutations are adding is beneficial. Could be akin to the dings and rust marks and dents you get on a car. But it is adding diversity, okay? And so you can even take it back to the Australopithecines. You can see here going back 7 million years ago. That's a lot of time where mutations are adding genetic diversity. And so you'd have, in the model, the evolutionary model, you'd have erectus apparently living on the globe between 1 and 2 million years. Maybe they exist in a population of 1 million. Okay. Perhaps their generation time is 25 years. That's a lot of genes floating around that population. We should expect higher levels of genetic diversity today. As a matter of fact, I want to ask Kat, where's all the bones? Where's the evidence of this so-called evolutionary history? You got Homo erectus living on the globe between 1 and 2 million years. Let's say they exist in a population of 1 million. 25-year generation time. Where's the billions of dead bodies? Where's the evidence for their existence? The campsites, the civilization, the grave sites, all the dead bodies. You could fit all erectus bones in the back of a car. That tells me that the evolutionary story is not true. Okay, so the separate ancestry model predicts low genetic diversity. The evolutionary model predicts high levels of genetic diversity. What do we discover? Well, humans are 99.99% the same. Any two human beings, nearly identical in their genetic makeup. And so that makes sense. From the biblical perspective, we'd expect low genetic diversity. That's what we see. We don't see the types of diversity, levels of diversity that if evolution were true, that we should expect to find. Do evolutionary rescue devices work? Out of Africa population bottleneck? We'll see if Catz appeals to that, and then we will address it. Mitochondrial DNA. Outside of the nucleus, we have the mitochondria. These are the powerhouse of the cell, as people like to hear. You probably know from biology class. These structures carry their own type of DNA, mitochondrial DNA. Now, mitochondrial DNA is inherited from the mother's side. It is uniparentally inherited DNA. And that kind of DNA, its function is it basically controls the conversion of sugars into energy. It creates ATP. And in each cell, you have many little, the number does range, mitochondria that produce energy for us to live and breathe. And that energy sustains life and body function. Notice this. This is fascinating, guys. Genesis 9:19. I definitely want to see Catz address this. These are the 3 sons of Noah, and of them was the whole earth overspread. So the Bible is making some very significant claims that every single human today has originated from 3 founding couples, Shem, Ham, Japheth, and their 3 wives. Those would be the daughters-in-law of Noah. Well, notice this. This is a mitochondrial DNA phylogenetic tree. This represents people. This represents individuals, people groups over time. The lines represent DNA differences or mutations. The tips are individuals. And you'll notice all mitochondrial DNA here can be traced back to 3 main haplogroups. These are not even contested by the evolutionary community. These are the L, M, and N haplogroups. Now, this family tree, going right back to 3 major haplogroups, is incredible because what we just read in the scripture is that all human beings today have descended from 3 reproducing couples. And since mitochondrial DNA is inherited maternally, these 3 major haplogroups right here in the middle, these are these 3 nodes, they could represent Noah's 3 daughters-in-law. And yes, they could live at the same time. They can be live contemporaneously because the lines separating L, M, and N, they're short. That suggests just a few DNA differences. There's only a few DNA differences separating these 3 nodes. The R haplogroup could be a post-Babel dispersal event, but the lines radiating out from them, they're longer, suggesting more mutations, more DNA differences over time, and post-flood history. Guys, the Bible demanded a single woman that every human being descended from. The evolutionary model did not predict this. When we look at worldwide mitochondrial DNA, the family tree collapses rapidly to a single person. This is Eve. The real Adam. The Y chromosome is a master control switch. It influences the expression levels of thousands of genes on all the other chromosomes, from the time a boy is in the womb to puberty and on into adulthood. The Y chromosome is also uniparentally inherited DNA. It is passed down on the father's side. And so, do we have evidence for one Y chromosome ancestor as well? The answer is yes. And we'll see that in a second. I do want to point out that in the mitochondrial DNA, the mutation rates are a lot faster than what's called the phylogenetic approach, what the evolutionists like to focus on. If we dig a little deeper, we'll get into the point that the neutral mutation rate approximates the fixation rate. And I got 5 minutes here, so I'll move quickly and then hand it over to Matt. And so this paper here from 2022 is making it clear that the observed mutation rate is approximately 10-fold higher than phylogenetically derived rates. Notice this from the Parsons paper in 1997. This is roughly, talking about the mutation rate, roughly 20-fold higher than estimates derived from the phylogenetic analyses. Again, 20 times higher. So what we see is low diversity, just a few DNA differences in the mitochondrial DNA, few DNA differences in the Y chromosome, but we have fast mutation rates. In the mitochondrial DNA, the rate is about 1 mutation every 2 to 5 generations. In the Y chromosome, it's roughly 1 to 3 per generation. And so with few DNA differences, low diversity, and fast mutation rates, what this is biblical time. One man, one woman. Notice this phylogenetic tree. This is similar to the one before. This was put together by Dr. Rob Carter, but there's some really interesting things here. So notice this, HVR. This is the most common haplogroup in Europe. You'll notice differing size branches. What this is telling us is that people in the same amount of time, some people are picking up more mutations than their cousins. So that tells us mutation rates are variable. And we have papers, 2022, showing 2 families, germline hypermutation. They're passing on even more mutations. Mutations in DNA repair genes can result in hypermutation. That means the evolutionists, they have a problem there because they're largely going to have to assume a strict molecular clock, a constant mutation rate, in order to take humans and chimps back to a common ancestor, roughly 6 to 9 million years. This is a Y chromosome family tree, same thing. We see people groups, we see individuals, we see one man, we see the reality that in the Y chromosome, we have few DNA differences. If I were to just draw a circle right here in the middle, we do have outliers that we can explain, but right down the middle in here, you're looking at about 200 mutations. Well, with a mutation rate of 1 to 3 per generation, again, that's biblical time. Notice this. This is a paper that just came out. This is incredible. 2025. If you look at the supplementary material, over 12% of the human and chimpanzee genomes represent what's called a gap difference. These are sections that can't even be aligned. So right off the bat, you're looking at roughly 88% difference in whole genome sequencing. And it's interesting because even in your similar sequences between humans and chimps, you have differences in the way that those genes are regulated or expressed. You have differences in alternative splicing, epigenetic differences. There's a whole lot of differences, and it's important. Remember this, and I want Catz to remember this. It's the differences that make all the difference, because Creationists, we can explain the similarities Evolutionists can explain the similarities, but the massive differences, that's where the evolutionists struggle, but the creationist expects. Here's an example. Here's the last point I'll have time to cover. This right here is an evidence that I believe precludes evolution and that Catz is really going to have to address. These are Y chromosomes between humans and the great apes. You'll notice right off the bat, the human Y and the chimp Y. The chimp Y is half the size of the human Y. The chimp has lost the entirety of its heterochromatic arm. This right here, it's all color-coded. The light gray, this is heterochromatin. It's this densely packed kind of DNA. It largely comprises what's called satellite DNA, repetitive sequences. It's not even present in the chimpanzee Y chromosome. And so when you consider overall architecture, size differences, and gene content, the chimp Y and the human Y are less than 30% the same. Actually, we see a break in the nested hierarchy where humans and gorillas in their Y chromosome, they're more similar than either is to the chimpanzee. That's fascinating. And this is 2024. It gets even worse for evolutionists. And Catz here tonight, unfortunately. We've got the most sophisticated DNA sequencing data for the Y chromosomes, the complete sequence and comparative analysis of ape sex chromosomes. You've got human and chimpanzee. You can scroll here and see less than 30% the same. Less than 30% the same. More than 30% of the chimp Y chromosome fails to align to the human Y chromosome. The evolutionary community are forced to say that portions of the Y chromosomes that they've been rearranged. We got gene loss, gene gain, and large-scale chromosomal rearrangements according to them. And so how do the evolutionists explain this data? We'll see here tonight from Conspiracy Catz or Culture Catz, and I'm excited to engage this data further. Matt, brother, feel free to hit unmute, and you've got 20 minutes. Share screen, you're good to go.
All right. We're going to start off with the age lifespan, because if these were real people and they're talking about real age limits, then we see in the book of Genesis that they lived for a really long time. And then after the flood, the lifespans dramatically diminished. Now, that's because of something called patriarchal drive, and it had to do with Noah. But what we're going to be focusing on is the lifespans, and we can see them in multiple cultures around the world. So it doesn't just fall into the biblical thing. A lot of people talk about this. Kind of strange that they would invent such a weird thing outside of reality when you want people to believe your religion right out the gate. But what do we know about genetics and the theory of aging? Well, your genes, as well as the mutations in those genes, are responsible for how long you're going to live. So there's a geneticist by the name of Dr. Aubrey de Grey. He's a biomedical gerontologist. He says that humans should live for about 1,000 years. But he's not the only one that said this. There's a book written on it by David Goldhill called Longevity, Why We Are Living Longer Today, and the Discoveries That May Allow Us to Live to Be 1,000. And then we see another molecular biogerontologist talk about how old we could possibly live. And an expert believes we could live for another 1,000 years. We see the same thing. Humans may live to be about 1,000 years. This is by Ray Kurzweil. He's a guy that spends about $2,000 every single month to get there with supplementations until we get the technology to live this 1,000 years. And it's true. You want to live for 1,000 years? Be careful what you wish for. How achievable is this? Well, it's achievable through the field of DNA repair. The mechanism, we are going to be trying to be using something called CRISPR-Cas9. It's a gene editing software where we can erase mutations and go back to the original sequence. And they've done this one time in a gene in mice and in fruit flies, and they call it the fountain of youth. And in mice, just editing one gene allowed them to live 23% longer. And Dr. Voltaire said with just a small modification of the genome, you can go from 2 years life expectancy to 100. So, is there evidence for Adam and Eve that could live for 1,000 years? Yes. Is there the genetic potential is still inside of us today to reach 1,000 years? So isn't it interesting that the maximum human lifespan estimated by different scientists perfectly land on the lifespan of the limit mentioned in the Bible? What are the chances of the evolutionary secular scientists discovering this and concluding that? So if these experts in their field of genetics are wrong, how and why are they wrong? And what are we doing when we're removing mutations that add lifespan? When we're removing them, what sequences are we mimicking? We're mimicking Eve's consensus sequence. This is the one that's been published in the peer-reviewed journals, and you can read it right here, called Mitochondrial Diversity in the Human Population, published by Dr. Robert Carter. And it's on Creationist website right here, where you can go and you can see the Eve's consensus sequence, and you can see that the gerontologists are using that sequence to mimic. So very interesting. So the goal of this section was to show that there is genetic evidence for long life potential, like the Bible describes, built into us, and it's still there, able to be unlocked through modern day gene editing sequences. I find that fascinating. But what about physical evidence? Again, we are going to go to what were predictions. What did we find? This is one is Jake Herbert by ICR, Institution for Creation Research. He noticed that there's greater longevity with larger adult body sizes and delayed maturation. We're all linked together somehow. So when we go back in time, we want to ask ourselves, is this true? I just ask AI. It says, yeah, slower maturation, reaching puberty is what that means, is often associated with longer lifespans. Well, what's the evidence for that? Easy. We can go to humans. When they reach puberty from 9 to 12, they live 70 to 75 years. When they reach it at 13 to 16, they live 80 to 90 years. And this is through dogs, mice, rodents. You can have AI answer it all. It always is the same thing. Very fascinating. So we want to ask ourselves, how about our supposed ancestors, which we would just say are people that were closer to the flood? Well, they reached maturity at a similar rate or slightly slower. That's with Neanderthals. How about Heidelbergensis? Same thing. It looked like they took longer to reach adulthood. How about bone evidence? Thicker bones live longer. Thinner bones live shorter. Smaller bones live shorter. So we see the same thing. How about bones in ancient archaic human beings? That's right, thicker and more robust bones. Heidelbergensis on this one. How about Homo erectus? Same thing. How about bones in Neanderthal? Same thing. We see the exact same criteria. So when we line them all up, look, modern humans have the smallest and thinnest of all of them. And when we compare them all side by side, we see that clearly our ancestors could have lived longer by with an evidence through thicker bones, just like we see in the scientific evidence today. Here's a jaw of an archaic human being right next to a modern day person. So if you want to see the studies that go into it, there's your ICR links for anybody that would be interested. And then we are going to jump into something called genetic boundaries. Now, this is really important. So pay attention here, because this is very fascinating. They analyzed barcodes from hundreds of thousands of different species, and they found a telltale sign showing that all animals emerged about the same time. Here's what their charts show. Here's birds, fish, moths. They all form, they all have low ADP, is average pairwise distancing. So they looked in the mitochondria, and they looked at the most conserved region, which is cytochrome C. And then they looked in cytochrome C to an even more conserved region called cytochrome C, the CO1 gene. And then, so they always look at the first 650 base pairs and go from there. And this is actually a paradox in evolution called the Lewontin's paradox. And it says that the species of vastly different population size exhibit the same similar low levels of genetic diversity. Why? Because not a lot of time has passed. So this is what those DNA barcodes look like. You can look at a species and go, wow, those are very, very different. I wonder if we can use those to determine relations and ancestry. And here's what one of the authors of the paper was talking about. He said, although it's easy to imagine humans passing through a bottleneck 170,000 years ago, it's hard to believe that exactly the same thing happened in all species. Did herrings really pass through an equally recent bottleneck, and anchovies too? I mean, that's a good question. What in the world does that sound familiar to anybody? What? Perhaps a global flood? What else? A global bottleneck could have wiped out all aquatic, all bird life, all animal life, all humans. Come on. They looked in Africa. They said, well, nothing out of the ordinary happened about 200 to 100,000 years ago in Africa. So that nothing there. Then they decided, let's go to the fossil record. Clearly, there's going to be evidence there. Nope. No trace of anything in the geologic record 200,000 years ago either. Young Earth creationists, we had a field day with this. Every organization talked about this study. That's how good it was. And we just loved it because it was the first time ever we have genetic boundaries separating things like the taxonomic boundary paradox, right? Taxonomy places categories and boundaries, but now we have those boundaries in genetics to even prove it. And this upset the authors of that study to the point where they had to add a disclaimer to the top. They say, no, this study's grounded in strong support of Darwinian evolution. I know some people out there say, no one believes Darwinian evolution. Well, They do. Anyway, including the understanding that all life evolved from a common biological origins over billions of years. This work follows mainstream views of human evolution. We do not propose a single Adam or Eve, and we do not propose any catastrophic events, even though they went looking for those catastrophic events. But the problem is that even the news press picked up on this, and they said what we know has debunked much about what we know about evolution of species today. So isn't that funny that they're debunking it in these articles when interviewed? Like right here, another unexpected find in the study was species have clear genetic boundaries and not much in between. The absence of these in-between species is something that perplexed Darwin. Right in between the fossil record, he expected, tons of intermediaries. Can't find them. The conclusion is very surprising, and I fought against it as hard as I could. That's what the authors of this paper said, but yet it strongly supports Darwinian evolution. So they say one thing after the fact because they didn't expect so many people would be like, wow, this is such great evidence for us. There's such great evidence for this independent line of ancestries that we're talking about. And, here it is with wolves. We provide several lines of evidence supporting a single origin for dogs, disfavoring models in which dog lineages arise separately from geographically distinct wolf populations. Exactly what the biblical thing said, right? It would be an unclean animal would only be going on by pairs of two. So we expected a convergence going backwards in time, right? We'd expect to converge on the consensus sequence that we evolved from, chimpanzees and humans. What do we find? The exact opposite. Modern humans have a very similar consensus sequence to the origin of some people today have no mutations, and we have a maximum of 10. Then we look up at Neanderthal, they ranged around 8, and Denisovan around 10 to 18, and then Heidelbergensis around 18 to 20. But here's the thing. When you look at those mutations, they're in the same place as the human version. The humans on top, Neanderthals mutated right below it. It's in the same place. And what that means is when you run the consensus sequence through even AI, take consensus sequence from a Neanderthal and a Denisovan and then revert them, and it matches the human consensus sequence. And that should not be the case if it was on the other side of the bottleneck. There's no way it would converge on ours. It would converge, it branches out from ours. That's the interesting thing. And how do we know that? We can see that when those mutations are removed, it matches our consensus sequence. And it would be the other way around if evolution was true. And that takes us to real quick, does creation model make any predictions about this? Yeah, matter of fact, Neanderthals were predicted way ahead of time by young Earth creationists, by them actually being just regular humans. And that was found recently, which is why humans and Neanderthal are less similar than polar bears and brown bears. And they even admit that humans belong to the same species could rewrite the history of evolution. And what else do we know about them? Well, they couldn't even have lived for over 10,000 years. How do we know that? Because of their population sizes. They admit that they lived around 10 to 15 people, including children. Some say no more than 20. So let's make a chart based on what that study found. They found that inbreeding with just 10 to 15 people allows a maximum lifespan, maximum 500 years of their existence. If they were between 25 and 50, they can only have lived up to 1,000 years. Well, how in the world could they have lived 400,000 years? It's impossible. And the ironic part is evolution has gotten every prediction it's ever made with Neanderthal completely wrong from its very beginnings. Matter of fact, they were called Homo stupidus in the beginning. So this brings us to our next question. When did this happen? So we found the genetic research going back into that very study because they just arbitrarily state 100 to 200,000 years. That's why they wanted evidence for it and went to the fossil record, couldn't find any. So the study authors speculated that somehow life nearly got wiped out across the board about 100 to 200,000 years ago and then restarted. So we want to know, when did that happen? Like, what is going on here? And that will bring us into mutation saturation. Over time, mutations build up in the mitochondria. It doesn't matter if they're neutral. It doesn't really matter if they're beneficial. It doesn't matter. They still are building up. They're still occurring. So when did the split happen? They used to say 5 to 7 million years ago with a maximum of 17. Well, Nathaniel Jensen from AIG decided, let's run those numbers on how many mutations should have occurred over that amount of time. You get an upward limit of 21,450. In reality, we only found 1,483. This is in the mitochondrial DNA, by the way. Not very many. I decided, let's check the CO1 gene. Over time, we've been separated from chimpanzees. About 65 mutations have occurred. So that means that over 7 million years, about 2,450 should have occurred. We're nowhere near that. How about bears? Bears supposedly emerged about 40 million years ago. They should have between 3,000 to 775,000, depending on 4,500 years ago. I did really a big favor by saying maybe they had a common ancestor just about 4.5 million years ago, and then or 40 million years ago. The range still not so good for evolution yet again. You can do the same thing with sheep and goats. Same thing, range completely off. Nathaniel Jensen ran it in flies, worms, water fleas, no matter where you look. How about whales? Same thing. Massive amounts of mutation differences should be there, and they're not. How about in chickens? Same thing, 129 actual differences. Almost 4,000 should exist. You know, evolution says, well, you know, the chicken came before the egg. That's what happens when you ask evolution, right? Science actually says the chicken came before the egg, kind of like the biblical creationists have been saying. Now, the biggest critic of all is named Evograd. He wrote a blog against Nathaniel Jensen, and here's his reply about that very study. He said, well, if that amount of mutations actually did occur, you would only be able to see 12,000, not the estimated 21,000 that he showed on that chart. The 12,000 decisions are still far too many compared to the observed number. I just think it's interesting that Jensen used these silly visual numbers. So don't worry, I removed those silly numbers and just left 12,000 on there. And actually what we found. So even the biggest critic, who's a geneticist, has nothing to say against this evidence. Like, it's literally just ignored entirely. So that's a heck of a challenge for anybody to try to refute. So then we go to, what in the world happens when you type in, how old are humans? You get 300,000 years. But why? Why is that? Why do they give that number? Does that come from the empirical evidence that we get from observed studies? There's only 24 fixed substitutions in the entire human population. No, it does not. It actually comes from calibration of the fossil record. It comes from nothing observed. And I had to double check with AI just to make sure for you guys. Sure enough, evolutionary biologists use the fossil record to calibrate phylogenetic trees. Why? Because Charles Lyell had a secret agenda when he created this a long time ago, and it was to free the sciences of Moses. He was an anti-creationist, and that was his goal. So evolutionary theory estimated about one substitution should arise every 300 to 600 generations. Well, what did we actually find? The very first study to actually come out with Howell, and he found one substitution every 25 generations. Way too fast. But it's okay, because he was looking at a family that might have had a optic neuropathy disease. So maybe just mutations were too fast because they had a disease. No problem. Along comes Thomas Parsons for the forensic FBI. He had access to the blood banks, and he has the ability to look at get blood from all different types of people. African Americans, Caribbeans, Sierra Leone, British Caucasians, Americans, vastly different. He wanted to get a substitution rate, and his results were the most recent common ancestor 6,500 years ago, landing on the biblical timeline. And he used 10 of the top geneticists of his time, and here's his coalescence calculation for anyone that cares on how he got it. And a lot of people say, well, he used the fastest mutation rate. Actually, no, he used the mid-range one. Most of them were actually even faster at one substitution every 17 out of, as you can see here, 73 lineages. So a lot of people go, oh, well, you're just looking at the fastest mutation rates. No. Matter of fact, one of his biggest critics named Holland decided to join him the following year and run the math again. And it was basically identical with one substitution every 30 generations instead of 33. So it actually was faster. So the FBI use his mutation rate today, and they track back and go into known history.
You've got 3.5 minutes, Matt.
Okay. All right. No problem. So what do we know then? I was going to go through more of those studies, but no problem. We have the observed rate way up here with the estimation rates of evolution way down here. They don't match. One's an assumption, one's viewed, one's falsified. All the top geneticists completely agree with this. Here's an evolutionist named Ann Gibbons, and she says that it's now worrying to evolutionists. She is one. She's using the term, so don't get mad at me. It could also complicate the lives of evolutionary scientists who say that the mutation rate should date back to the evolutionary clock, you know, and evolutionists are most concerned about this faster rate. And why? Because using the new clock, she would be a mere 6,000 years old. So, of course, they invoke, well, you know, this is 10 times faster, and there's no evidence of why this should be the case. They've checked for mutational hotspots, selection, random genetic drift. One person decided, I'm going to run just selection to determine. Maybe selection is what's really slowing it down. He came to the conclusion that, nope, selection is unlikely to be the major cause for this. We just can't figure it out. You need rapid selection. Well, it's unrealistically high for beneficial mutations. You can't get it. Even Kimura back then said it. So if all these experts in the field are wrong, why are they wrong? I mean, they admit that in general, there's a perception and a competition to get an older date, and there's kudos in it. That's why they're trying to push the rate back. It's landing on biblical timelines. Evolution theory is based on mutations. So if mutation rates are fast and they require it to be slow, that changes everything. The predictions are completely out the window. The FBI uses them. They match historical dates. They're used by young Earth creations to make predictions. They're found in all animals, insects, reptiles, and birds. So you can't say, well, the most recent common ancestor, of course, it's not the first human. Well, now you have to account for why it also lands in all animals and insects and birds that also go back and land on the biblical timeline as well. Good luck trying to do that one. And we also, here's a chicken study. 15 times faster. The substitution rate in avians estimated based on fossil calibrations. What did they find? 15 times faster than they were, just like human beings. 50 generations back, super deep pedigree, 2 different lineages, fast mutation rates. Selection had no effect on them whatsoever. Population growth rates converge on the biblical timeline. Legends of a global flood everywhere. Animals, birds, all have the faster mutation rate. Substitution rates and coalescence point using pedigree studies converge on the biblical timeline. Fast mutation rates and low genetic diversity is a paradox for evolution. It's not for us. It's totally expected. There's no logical explanation why millions of people living on islands uncontacted with human beings 6,000 years ago would have completely died off. And when you ask AI the same thing, it says 6,000 years ago, you completely need a bottleneck. Matter of fact,
Matt,
yes,
you've got about 10 seconds, but if you need an extra 5, that's okay. Feel free to finish your final thoughts.
I don't need to. That's good. That's all right.
Okay, Matt.